Imagine hiking when your bottle runs dry. Someone remembers a survival show and points out that your body is still producing liquid. Then comes the famous suggestion: drink your own urine. It sounds disgusting, but may be possible. Now compare it with the other material your body throws away. Nobody calmly recommends eating poop. If urine and feces are both body waste, why do they feel like completely different risks? The short answer is that waste is the wrong category. A small accidental sip of urine is often less infectious than swallowing feces, but it does not make urine safe or useful as drinking water. The real difference depends on where the material came from, which microbes are present, how concentrated it is, how much enters your body, and who is exposed. To see why, we have to follow two very different routes through the body. Your body discards carbon dioxide through the lungs, sweat through the skin, urine through the kidneys, and feces through the intestine. All four are called waste, yet breathing somebody's exhaled air is not the same event as swallowing their stool. Waste only means the body is done with it. It says almost nothing about infectious risk. Think of two trash bags. One came from an office wastebasket. The other came from an isolation room. Both are trash, but their contents, pathway, and handling rules are different. Biology works the same way. Risk is better described by 5 questions. What is the source? What organisms are inside? What is the dose? Where does it enter? And how well can the exposed person defend themselves? Urine and feces begin answering those questions in different organs. Blood enters the kidneys through branching vessels and reaches about 1 million tiny filtering units in each kidney. In each unit, a glomerulus lets water and small molecules enter a tubule, while cells and most large proteins stay in the bloodstream. Then the tubule performs the clever part. It returns most of the water, minerals, and nutrients that the body still needs, while moving additional wastes and excess acid toward the urine. What remains travels through the ureters to the bladder. That means urine is formed from filtered blood plasma and selective reabsorption. It does not pass through the colon's dense reservoir of intestinal microbes. That difference usually gives fresh urine a much lower load of gut pathogens than feces. But lower is doing important work in that sentence. The old claim that healthy urine is sterile is wrong. Modern methods detect microbial communities in the urinary tract, and a voided sample can pick up organisms from the urethra, skin, or nearby genital surfaces. A urinary infection can add far more bacteria, so the kidney pathway explains lower average infectious risk, not guaranteed cleanliness. Another common argument says urine is mostly water. That can be true and still be misleading. Seawater is mostly water too. What matters is what is dissolved in it and how concentrated the solution has become. Healthy kidneys can make urine extremely dilute after heavy water intake, or very concentrated during water deprivation. Human urine can span a very broad physiological range of osmolality. After hours without water, it may become about 4 times as concentrated as blood plasma. The darker sample produced during dehydration is exactly the sample a stranded person would be tempted to recycle. Drinking it sends water back in, but it also returns urea, salts, and other dissolved material that the kidneys were removing. It can also return medication metabolites or contaminants. Repeating that loop does not create new water, and United States military survival guidance specifically says not to use urine as a water substitute. A tiny accidental sip and deliberate repeated drinking are therefore different questions. One may cause no obvious problem. The other is an unreliable hydration strategy with growing disadvantages. Feces take the other route. Food is digested and absorbed as it moves through the stomach and small intestine. The remaining material enters the large intestine, where water is removed, and a vast microbial community helps process what remains. The colon is not a dirty pipe. It is one of the densest ecosystems known, with hundreds of billions to roughly 1 trillion microbial cells in a milliliter of contents. Many of those organisms are normal partners. They help break down compounds, interact with the immune system, and occupy space that might otherwise be taken by harmful invaders. Stool is therefore not simply undigested lunch. It contains water, fiber, and other residues, shed intestinal cells, microbial products, and an enormous microbial biomass. Reviews have estimated that living and dead bacteria together can make up roughly 1/4 to 1/2 of fecal dry solids. Most of those microbes are not trying to hurt you. The problem is that an uncontrolled sample can also contain disease-causing bacteria, viruses, or parasites, and there is no label on the outside telling you which sample does. This is where dose changes the story. A person infected with norovirus can shed billions of viral particles in feces, while only a very small number may be enough to make another person ill. The dangerous serving size may be too small to see. The pathway is equally important. A microscopic smear moves from feces to a hand, from the hand to a door handle, from the handle to food, and from food to another mouth. Or sewage contaminates water, and the water carries the same route at a larger scale. This is the fecal-oral pathway. It is not one bizarre person deciding to eat a visible pile. It is usually an invisible chain of transfers. Your stomach acid, intestinal barrier, resident microbiome, and immune system all provide defenses, but no defense is absolute. The outcome depends on the organism, the dose, and the host. Children, older adults, malnourished people, and people with impaired immunity can face more severe consequences. Someone may also shed a pathogen before symptoms appear, or after they feel better. Smell, color, and confidence are not diagnostic tests. That is why I Feel Healthy cannot certify feces as safe, and why washing hands with soap matters so much. Soap and friction do not negotiate with grossness. They interrupt the physical route. There is an apparent contradiction here. If the colon contains trillions of normal microbes, and many of them help us, why is material from the colon treated as dangerous? Because location is part of biology. A community adapted to the intestine belongs behind the intestinal barrier, inside one particular host, in competition with thousands of other species. Move an uncontrolled sample into another person, another body site, or a damaged host, and the rules change. Even a sample dominated by harmless organisms can carry one unwanted pathogen, and a pathogen does not need to dominate the sample when its infectious dose is tiny. So feces are not dangerous because every microbe inside is evil. They are dangerous to ingest because they are a sample that has not been screened, with high microbial biomass, from the exact system through which many enteric pathogens leave one host and reach the next. Now for the strangest part. Doctors sometimes transfer carefully prepared fecal microbes into a patient's intestine. Fecal microbiota transplantation has been used for selected people with recurrent Clostridioides difficile infection, a condition in which the normal gut ecosystem has been badly disrupted and a harmful organism keeps returning. This is not a patient eating random stool. Potential donors are screened, material is processed, the indication is specific, and delivery happens under medical supervision. The goal is to restore an ecological community that can resist the recurring infection. The procedure proves an important point. The category feces is not magic poison. Context can change biological material from a hazard into a treatment, but the controls are the reason that transformation is possible. The United States Food and Drug Administration has issued safety alerts after serious infections were linked to pathogens transmitted through fecal microbiota products. Screening reduces uncertainty. It does not make do-it-yourself exposure sensible. That medical reversal brings us back to urine. Saying that one exposure is lower risk than another is not the same as declaring it safe, useful, or therapeutic. For a healthy person, a tiny accidental urine exposure often passes without illness. Risk rises if the urine comes from someone with a urinary infection, contains blood, has been contaminated after leaving the body, or carries substances excreted from medications. Repeated drinking during dehydration adds a concentration problem even when infection is absent. Fecal exposure deserves more caution because the pathway naturally carries a much denser and less predictable microbial load. If exposure is followed by persistent diarrhea, blood in the stool, severe vomiting, fever, or signs of dehydration, medical advice is appropriate. The most severe danger from diarrheal disease is often the water and electrolytes lost afterward, especially in vulnerable people. The practical prevention is unglamorous because it works. Safe drinking water, adequate sanitation, clean food handling, and hand washing with soap. These measures are not rituals of disgust. They break a transmission system. Return to the hiker with the empty bottle. The urine suggestion was never a clever loophole. Urine is not sterile, and it is not emergency drinking water. Feces are not food, even though many gut microbes are normal, and some can be used in tightly controlled medicine. So the memorable rule is simple. Do not drink urine, do not eat feces, and do not confuse less dangerous with safe. Both materials leave the body, but they arrive at the exit by different roads. When you want to understand the risk, follow the route, count the dose, and consider the host.